Hello everyone, in this video we're going to talk about DNA isolation, especially plasmid DNA isolation. This occurs in a few important steps and we'll start by comparing it with just genomic DNA. Based on the composition, what we already know is that plasmid DNA has a circular double-stranded form while genomic DNA is usually double-stranded linear. And during our normal DNA extraction, which includes cell lysis, removing of proteins and RNA, and some centrifugation to elute the supernatant, which is going to be the low molecular weight DNA, we might end up with a solution that is going to have both linear DNA and plasmid, because these steps are actually not too specific to actually separate between the two type of nucleic acid. And when it comes to plasmid DNA isolation, one of the earliest technique that was actually discovered on how to isolate was known as the alkaline lysis technique. This is because normal genomic DNA extraction uses usually an enzymatic using enzyme or a mechanical way of lysing the cells. What do I mean by this? So one is going to have two test tube like this. Let's say if you're performing enzymatic and mechanical here, with your mechanical you are going to have a sample of beads like this, very tiny beads, and your DNA is going to be here in a solution, and all you do is vortex or shake this in such a way that the cells are going to break like this, and you're going to end up with your DNA, and you move to this step up here, which is centrifugation. With enzymatic, it was more of putting an enzyme, and this enzyme, based on its property, it's going to break the cell and eventually result in the same type of output. But with alkaline lysis, this is a method in molecular biology where plasmid specifically is isolated, or other cell components such as protein, by breaking it open, because it's a lysis anyways. But how do we do that? Because the first thing is using now a detergent such as sodium dodecyl sulfate and a strong base such as sodium hydroxide to actually increase the pH of our solution. And once the pH has increased, as we see in this graph picture that we have here, usually it will increase within a range of 12 to 12.5, and that increase in pH is going to cause the double-stranded linear piece of DNA to denature. But it shouldn't be done for too long, because although alkaline condition denatures DNA, but we don't want it to denature our plasmid DNA, because that's what we're interested in. Then this is going to be followed by a next step, which is now neutralization. So we move from a strong base solution to a neutralized solution, and the way the solution is neutralized as well is using acidic sodium acetate to bring the pH down. Once the pH has been brought down, what one may notice is that the single-stranded linear DNA that has been denatured is going to aggregate, and if they aggregate in the solution, they are going to have a high molecular weight. And because they're still in a solution like this, now all we have to do is centrifuge like it was done here. And once we centrifuge, these aggregates pellets are going to actually sink at the bottom because of the high molecular weight, and our supercooled plasmid, which carries a very relatively low molecular weight, are going to be the supernatant, and we are going to take them and purify them and carry them for further analysis such as recombinant technology. But the bottom line is plasmid extraction involves weakening of the bacterial cell walls, lysing the bacterial chromosomal DNA by increasing the pH, which denatures the DNA, but we don't do it for too long, because once it has been for too long, we're going to lose the plasmid as well. As soon as it has been done to a point where the linear DNA has been denatured, then we move into neutralization, and we're going to separate our aggregate from our supercooled DNA. So that is in a nutshell our plasmid DNA isolation is performed. But nowadays there are a couple of cool inventions, and we also use kits, most likely in labs and all that. But this was the initial principle behind our plasmid DNA isolation. I hope this helps, and good luck.